Once again, thank you very much for the invitation. I'm very happy to be speaking to the audience of people that we've been reading papers from. It's nice to, well, see you and hear you, at least virtually. So my name is Maciej. I am a laboratory technician at the moment at the Medical University of Lublin. This is Eastern Poland. And right now I'm working on my PhD, which is also interconnected with the work that we recently published and which we were invited here to speak about. So first of all, I would like us to introduce ourselves more. I just started that we are a cooperation of two teams. One is from Medical University of Lublin. This is in Eastern Poland. And I'm part of this team. This is a team of Krzysztof Jóźwiak. And another part of our team is a team from Manuela Bartolini's lab from University of Bologna in Italy. And I have to single out one other person, which is a first author of the paper that I'll be describing here. And he comes from Professor Bartolini's lab, but he changed his occupation to a National Research Council of Italy, also in Bologna, Daniele Tedesco. I think he's here, so for more like complicated questions regarding the chemical nature of this research, I think we could ask him. So what make us work together? So a common interest really, which is a medicinal chemistry, which might be a bit different background from where I presume most people working with SMALPs came from. So we are neither membrane biologists, like sensu stricto, and we are not structural chemists either. So the Polish lab deals is an interdisciplinary team dealing with molecular mechanisms of drug-protein interactions, and we are made from chemists and biologists. I'm myself a biologist, so we deal on a different levels from starting on in silico and ending in vivo on animals, and mainly all of in between these levels. So the in vitro study of this ligand protein interactions, and we are mainly interested in membrane proteins, and we are interested in GPCRs. We've dealt with ligand-gated ion channels and with nicotinic channels. And Italian lab is a mainly chemistry, pharmaceutical analysis and bioanalysis lab, which also develops methods for studying drugs in vitro. I think it's fair to say that recently the main target was Alzheimer's disease, and especially multi-target ligands. So why did we want to use SMALPs is because our end goal is to have, or let's say, adapt an experimental platform for affinity binding, especially with use of ITC for studies of different membrane proteins and ligands information. ITC is a quite particular technology, the method, which shows the full thermodynamic information about the binding, but it needs water-soluble homogeneous sample. And it's also, on a side note, both of our labs are interested in more subtle parts of this interaction, meaning we're interested in stereoselectivity of ligand binding. And that's one of the reasons to use this method because it allows to detect these subtle differences with binding. So we had different projects in cooperation ranging from 2015, which were financed by Polish National Agency for Academic Exchange, also the Italian Ministry of Foreign Affairs. And part of this work led us to publish our first paper in BBA biomembranes, first paper in SMALP field, which were focused on the trials and tribulations with trying to get SMALPs to work with low-yielding cell lines, especially for isolating of GPCRs. And I'm here to speak about this paper in particular, because I was invited by you. So, as I said, we are interested in different GPCRs. Some of them have quite obscure pharmacology, but we wanted to start from something that is known, something that could act as a model. So we settled on a beta-2 adrenergic receptor, as painted beautifully by David Goodsell here. And what are the reasons? So it's probably up to date. It's fair to say that up to date, it's the best structurally described human GPCR, with the first structure crystallized. Yeah, this was the first human GPCRs were crystallized and structure published. So we know a lot about ligand binding pocket of this receptor, and which led us to have a multitude of different ligands and functional tests that might be used. So it can act as a good model system. And despite all of these advantages, this whole system regarding the regulation of it and intracellular signaling is quite complicated. And it's also heavily dependent on cellular context. So we really want to have this context preserved during our ligand binding studies, which pushes us towards the expression system that's is the most similar to human organism, possibly. We know for sure from literature that the context of the membrane is very important for these receptors and maybe generally for other GPCRs. So, for example, in 2008, there was revealed there is a cholesterol consensus motif. And I think the first crystallization a couple of years earlier was possible only with a cholesterol. So we know that the sterols play a role in the functionality of this receptor, but we also learned that the lipids, especially the lipid head groups or charge, might make a difference into the functionality of this receptor. In particular, certain lipids stabilize different conformations of the protein. So they act basically as allosteric modulators. So we have a good established reasons to have it a very native system for that. So what are our options? So we don't have many really when we looked at it, because probably the SMALPs are the only one available that might let us avoid the detergent solubilization stage. And as we know, the delipidation of the membrane proteins is a main reason for losing the functionality for the above mentioned reasons. And SMALP, when it appeared more widely in the literature, it made us open our eyes, and that might be for us because we don't use detergents. We preserve the annular lipids near the protein, and it seemed like would be quite ease of use. Okay, so just briefly, we using the papers, the Gulati ABC transporter paper from 2014 and the Nature Protocols from great paper from Sarah Lee. We based it, our initial protocol, heavily on that, which is like a quite high concentration of SMA, which will be later lowered to 4% with an incubation and later ultracentrifugation. That was our SMALP preparation. And for the purification, we wanted to go with immobilized metal affinity chromatography with later purification of fractions by SEC. But first, we had to choose our expression system of choice, which in our case was HEC-293. Reasons being, they grow fast, they are quite robust, they are not picky about the medium too much, and they are easy to transfect, and they have a high expression of protein. We're able, though, to culture them adherently, and this poses some problems with efficiency just at the start. So first of all, like 10 of these dishes, we had much more, gives around 1 gram of wet cell pellet, which gives around 70 milligrams of raw membranes. And that was our first hurdle. Second hurdle was we knew we would have other efficiency problems that we will have to address at some point. So one of them was expression efficiency, then the solubilization efficiency, which might be different for different SMAs, and then the purification efficiency that depends on the scheme. So we started right away from the scratch we wanted to do it right the first time. So we purposefully designed a recombinated human beta-2 adrenergic receptor. The scheme looks like that. So there are a couple of parts here that I'll explain. So this is the cleavable signal sequence of hemagglutinin. So based on literature, this signal sequence sends the protein onto the membrane, even though the first transmembrane part of the GPCRs do that. So thanks to this cleavable signal that's being cut off during the expression, it enhances the expression on the protein on the membrane. So we chose a flag tag on the N terminus of the protein in order to have means for immunodetecting human beta-2AR, obviously, attached with a TEF site, which is cleavable with the use of a particular enzyme. We have this strange histag. So we decided on it because reportedly it more robustly attaches to surfaces. So somewhere far away up on the road, maybe there will be SPR going on. So this is 10 histags separated by this spacer and another 6 histags. It's maybe a bit longer, so that's why we have a TEF site to cut it all off when we need it. We cloned it into mammalian expression vector, which is armed with a strong enhancer and promoter for efficient expression, obviously with an appropriate selection genes. So then we went into the selection of the clones. We have chosen only the correctly incorporated inserts into the plasmids. We have excluded the clones which had like ambiguous. We obviously sequenced the whole insert, which had ambiguous base calls here, and we transfected it using polymer, cationic polymer. This picture is a control transfection which was made in parallel with a M-cardinal fluorescent protein to just control for the transfection conditions. we didn't want it to attach any fluorescent protein to our GPCRs, specifically for the reasons mentioned before, to have it as native as practically possible. So I'm just putting it up to show the to show the transfection efficiency. Then we did immunodetection to check whether we have both of our tags visible. Using dot blot here, we can see with a positive and negative control for both of the both terminal tags. Okay, then comes the moment of polymer selection. So we were kindly given by Dr. Stefan Scheidler from Polyscope Polymers from Netherlands two commercially available polymers with a commercial name Xiron, which will be that X30 and X25. So with different parameters, slightly, one is 3:1 styrene to maleic ratio, the other is 2.3:1. But also, I didn't mention, we don't synthesize our own ligands, but we have a wide cooperation with others. So we thought, let's maybe try and do our own polymer. Thanks to a cooperation with Rita Mazzoni's lab from industrial chemistry in also University of Bologna. So we aim to get something similar, just to see, maybe to have more of this polymer. It turned out to be a bit different, a bit higher mass, and we dubbed it C30. So we planned to stick to just one polymer, and but first we wanted to check the polymers itself, and arguably just the polymer solubility, not solubilization, but that is solubility, is an important factor which can predict the further efficiency of solubilization. So we checked with dynamic light scattering using 1% SMA solution in potassium phosphate buffer in different conditions. So we checked different pHs, and we can right away see the differences between the medium diameter calculated by DLC. So obviously we know as an amphiphilic polymer, it will aggregate in certain conditions. So we were not surprised, but we just wanted to choose like optimal conditions for solubilizing this polymer, because only solubilized polymer will be an active one for solubilization of membranes. That's what we figured. So right away, it seems that our polymer is significantly worse. So there might be different reasons for that. For everybody that's interested, I can point into the supplementary materials in our publication. There are more information about this particular polymer. So we have settled on pH 8. Based on that, it seemed like a optimal condition for that. And with a pH 8, we also checked ionic strength of the buffer, which turned out to make a bit of a difference even for our polymer. So for the further assessments, we went with the zero salt. So let's move further. Let's try it on cell membranes then. These are not yet the cell membranes with overexpression. These are the wild type HEC293 membranes. And we suspend our membranes in the buffer based on Sara Lee paper, Tris and glycerol. And we check the turbidity, the drop of turbidity in time after adding our polymer. This time it was at 1%. And we monitored it. And right away, we could see the difference, even though our polymer and commercial polymer are the same ratio, the difference is staggering. Might be the amount of free styrene or the different sequence. But also we see a huge difference between the solubilization efficiency. This was with the recommended 500 millimolars of NaCl. But when we dropped the salt content in the membrane suspension buffer, we see a much different picture. So we see an immediate drop of the turbidity right away, and even the X25 polymer with a 3:1 ratio became much better, also with ours. So, but based on that, we just settled on the one. So we went with X30 here. But we were surprised a bit about our findings, because looking at the papers, we seem that it's a bit different to what the literature says. So we did a bit of data mining in literature. It's just an illustration, because we looked through all these papers ranging back from 2014, around 50 publications that give their methodology. And in 90% of these publications, the buffer used for membrane suspension had a quite high salt content. But also only 10% of these papers have been working with the mammalian cell lines. And it's fair to mention a very nice Adrian Kopf paper that dealt with a rational optimization of the conditions. But very nice paper, but also done on the Escherichia coli, so or coli. So he also gives a similar figure to this. Maybe, just maybe, the cells used really make a difference to how it should be handled using SMA. So armed with that knowledge, we knew that we will have to use a SEC, size exclusion chromatography, for further purification of the membranes. So we knew that mostly it's used with a running buffer with a similar amount of salt. So we knew that it also might introduce our salt and contribute to some aggregations of the polymer and maybe SMALPs itself. So we decided first to check just the SMA solution in a potassium phosphate running buffer. And what we have learned is that with salt, we have a quite evident widening of the peak, and also going past the exclusion limit of the column. And we also learned, which confirmed our findings by DLS, we could see it also in SEC, that depending on the salt content, we can see aggregation of the SMA. So that was a nudge in a direction that maybe in SEC we also shouldn't use the salt. So we tried it on the membranes, wild type membranes with X30 SMA again. So we made SMALPs generally and ran them on a SEC. And here we have a very evident difference. So what we can see here, our interpretation is we can see a co-elution of, when there's a high amount of salt, there's a co-elution of free SMA with SMALPs. That's why it contributes to the increase of this peak and decrease of this peak, which is suboptimal. And also we are looking probably at the aggregates of the SMA. So this was in SEC running buffer. We decided to go with no salt again. And we checked this again on the wild type membranes, changing the suspension buffer, which the membranes were suspended after isolating them from the cell pellet. And we also see like a quite interesting view that the less the salt it is, the the better definition of peak of our SMALPs we can see in the range of elution value of the SMALPs. Okay, I think we were ready to go with the target membranes then. And we could see a very similar picture like with wild type membranes. So you could go as far as to say that it doesn't, in the apparent protein density on the membrane doesn't really influence the efficiency of the sec. But again, it's beneficial to have less salt in the membrane suspension buffer, as well as in the running buffer. We also did another one check because we all the time we were worried about the efficiency and the yield of our preparation. Unfortunately, for the ITC, we need quite high amount of protein. So we also checked on final concentration of SMA, and as also Adrian Kopf has showed in his paper, we figured out that we need much less than we thought initially. So we could drop it to 0.4 without the loss of efficiency, and definitely 4 is just too much because we also can see coelution. And the next point that we had to address is another step of purification, which was IMAC. So we started with our gravitational IMAC columns, which are, very cost effective. They are quite robust, but we kept using high volumes. We kept diluting our samples quite much, which is less than ideal in our situation. So we said, can we do it some way better? So we came up with an idea of using the HPLC system and packing a column with a relatively small bed volume with nickel agarose and trying to put it in line with SEC. Just minimize volumes and minimize losses and avoid also diluting the sample. So our setup looked like that. We have used an additional switch that allowed us to include or exclude an IMAC column in the system when we need it. So when the switch is in, let's say, position A, the flow of the running buffer goes like that, from HPLC pump through the SEC and straight to a diode array detector. So this is the resulting chromatogram that we could see. So using this setup, and we decided to designate a certain volume, which is our SMALPs from 12th to 19th milliliter of elution volume. And the switch allowed us to turn on the IMAC column only at the moment when it's needed. So we allowed the bigger particles, like maybe some left out membrane parts, to flow through this setup. And then after turning on the switch, the flow is slightly different. Still goes through the SEC, but then goes through the IMAC column. And in the end, it effectively loads up the IMAC column with only the particles that we're interested with, because neither we're introducing bigger particles nor small ones, which is the free SMA, which, as we know, it interferes with the IMAC itself. So it looks like that. This peak here, inverted peak, is an artifact of switching and the pressure drop, but it's irrelevant. what's important is what we can see is a drop of a peak here, which means that we have a retention and absorption of our SMALPs on the, I remind you, having a haystack to the IMAC resin. But next, our system might be used further for elution. So we switch up the direction of the columns, which we end up with something like that. We can now elute our SMALPs. So again, by the way, I didn't tell that. Then we can, of course, we can switch, and we did that. We switch again. That's quite crucial. We switch again at the 19th milliliter volume back to this system. So all of that goes, all of the particles that goes in this high elution volume, so all the smaller ones, don't go into the IMAC column anymore. So we have our loaded column. We switch up their direction, and the flow is as shown. We can now elute also with using phosphate buffer, but with imidazole. Direction of flow is like that through IMAC, again through the sec column, and through the detector, and we can collect the fractions here. After the first 5 milliliters of imidazole, we use a running buffer later using the same setup. And what we end up is this profile. Again, we can decide on the elution volume that we want to catch in our collection fractions. And then we could switch off and effectively exclude the IMAC column after we elute our SMALPs. And after switching back the direction of flow is like that. What does it accomplish? Well, we get rid of the imidazole because for standard protocol, we would have to do an ultrafiltration or a dialysis, which is less than ideal when you're dealing with low efficiency systems, let's say low yield systems. Imidazole also absorbs UV at 280. It might also contribute to the aggregation of the particles. So without doing, thanks again to this setup, we can get rid of the dialysis and or ultrafiltration steps. So did we end up with SMALPs? So we can use our tags for immunodetection. So this elution fraction we checked using a dot blot. So why dot blot and why the time is so high? So the bad news after all of that is we still have a very small amount, so that's probably not enough to do any functional studies. And this sample was later ultrafiltrated to concentrated. It was also added to a dot blot multiple times, and also the detection time was long, even though we had strong standardized anti-tag antibodies. Its still signal was very faint, but evident. So from this elution range, we definitely saw some, I don't know if you can see this, but yeah, there was a faint signal here from the flag. And from the haystack, we knew that we have both of our tags here. And we also checked other elution fractions just to see if we're doing okay, if it's pure. So we checked this elution fraction, 21 to 23, and we saw nothing. Again, another fraction, we saw again nothing. To confirm that, we also checked just the material let's say, raw SMALPs before purification. That's the sample that we put onto our chromatographic system. So we easily detected the tags, and the same in the raw membrane suspension. Also have a control to check whether our system is looking okay. So in the end, we ended with SMALPs, but as you can see, it needed from us a lot of optimization. And yeah, so we it's still difficult thing to do, to use a native system as we did, to get and use SMALPs for it and have enough protein for functional studies like we would like to, without resorting to some massive scale-ups in cell culture, like, I don't know, wave bioreactors, which are not available to everybody. And that's why we think that it should be the optimization of the solubilization conditions should be optimized further and should be explored as it was in the Kopf paper. I think it should be, we think it probably should be explored in different biological materials in different cells, because it seems it might make a difference. And depending on what the membrane context is. And last but not least, we hoped with this paper that we might help some people that, like us, wanted to start from the scratch, especially if somebody deals either with low yields or the materials that give a few amount of cells. It might be later beneficiary even for people that use SMAs, maybe from material from particular organisms. And we hope that the trend of optimization in the field will continue, because it might allow to use the SMALP technique for other uses than structural ones. So yeah, I thank you for your attention, and if you have any questions, we'll be happy to answer them. Thank you.
Thank you so much. Feel free to ask your questions if you have any. Just unmute yourself and ask your question. So I have one question for you. I mean, your target was very novel, or you had seen some like ITC experiments or like SPR experiment with your target in the literature using detergents?
I'm not aware of that.
Because maybe you don't need to, you don't have to use SMALP for...
Yeah, that's a very good question. And as another researcher from the dealing with amphiphilic polymers, Jean-Luc Popot, he always says that there's no silver bullet, and that it's if protein works in a detergent, it works. So it's a really good question, and it's arguable whether we should go in this direction. But we wanted to, you know, maybe go straight to the best conditions that we thought will be good for our receptor, because maybe detergents would be enough. But again, we're stumbling upon the problems connected with the lipidation of the receptor. And again, using the detergent would mean also optimization, because every protein is different. We used beta-2AR for this one as a model, but we're only slightly interested in this receptor pharmacologically. We're interested in other ones which are less known, which are not as well described, and would need from us to do an optimization on detergent selection anyway. So we said, why not go for this solution, which seems the best one in the field, but it turned out to, it still needs optimization.
That's correct. So you are trying to scale up your expression system, right?
Yeah, we will try to scale up our expression system at some point, but to contradicting a bit of what I said a second ago, in parallel, I am thinking of going to Pichia for that. And it's an ongoing work at this moment to have a construct ready, because this is a proof of concept, this our paper, and again, we prove to ourselves we are able to make SMALPs. Whether we are able to use it functionally, maybe not. Either we need to scale up expression systems very much, or bow down and try to check it on another expression system. So we'll try maybe go both ways and see where it leads us. But I remember because there was a conference in Leeds based on SMALPs. So we went there with Daniela, and someone told us that we're going into a very rough trip trying to express GPCRs in mammalian cell system, which proved to be true. But yeah, we learned a lot, and maybe some people will also learn. So
excellent, thank you.
Thank you.
So the length of your new polymer, I mean, is called C30. How long was that? I mean, what's the length of that polymer?
So it was the length. Okay. So what I shown you, I shown you that. Can you see it?
Yeah, 12 kilodalton. Yes, close to 13. Yeah. Okay.
Yeah. So that's how it turned out. So there were a couple of yes.
Go ahead, please.
Yeah. So there were a couple of problems with this polymer, and we had problems with hydrolysis of this polymer, and it was in a made in a different way, because these ones were the commercial Xyrons were delivered in a form of a very high, I think 20 or 30% of aqueous solution. So it's almost like a glue-like consistency, viscosity, and but it was in a salt form, and we had a free acid form, which might have contributed to the behavior of this polymer. And in the meantime, we also learned that, well, I'm not a chemist, maybe that's obvious to chemists, but either you can control the sequence of the polymer or the length. And it's very hard to do both at the same time, as far as I understood. So that also might have contributed to our polymer not being the greatest at solubilizing, despite having like a similar properties at the first sight.
Yeah, I understand. Yeah. But you didn't try SMA 2000. Most of the time, and I had two mammalian proteins to work with. And that's interesting that the final concentration of SMA that you use was 0.4%. I also used to use 0.5% for mammalian source, for any protein that I extracted from mammalian source. That is very interesting. So, but the amount of salt that I used was 150 or sometimes 100 millimolar. So that is very interesting. That kind of they have some sort of, our protocols are kind of overlapped. At the same time, the SMA, I used to use SMA 2000 for most of my solubilizations. So, I mean, if you have your hands on SMA 2000, that is also a good to go, a good choice to go. Yeah, maybe you get higher yield with SMA 2000. I'm not sure. You never know until you try, because literature in this field is a little bit inaccurate. So, but mainly SMA 2000, I used to get better results with SMA 2000. So this is something that you might try. Any other questions?
Hi, this is Ting.
Hi.
Very nice talk.
Thank you.
I have a question about the pure. So you did the SEC and also IMAC to do the purification, and then later use the immunodetection to test if the protein was there. My question was, do you know how pure the protein was in your sample after elution? Did it contain like also contain small amount of other things?
That's a very good question, and we don't know exactly. The amount of our sample that we end up was minuscule, just enough to check the presence of our protein. So judging from the our purification scheme and our chromatograms, I would say it will be a very unstrict term. It should be fairly clean, because we basically did like 2 steps of size exclusion, and we did and in the middle we did 1 step of IMAC, right? So and we did sec before and after the IMAC. I would really like to produce a more amount of, and we'll be doing that now, having the stable cell lines, and maybe trying to move it to a suspension. I know that people done that on hex cells.
Yeah. So based on this, how many like plates you used for getting these results for the culture? So you use like 15 centimeter plates, right?
Yeah, it was 15 centimeters plate, and for this it was, I think it was 40 plates.
40? Okay.
Yeah, and they were grown, and they were grown for a couple of weeks, and we based mainly on the transient transfection. This also gives a higher expression.
So how long you get the you did transfection, and when did you collect the cells? Like 2 days after transfection?
Yes, yes. For we aimed at 48 hours.
Oh, 48 hours. Okay. Thank you.
Thank you very much. Referring to the question about the remark about the polymer. So we also got we got a sample of DIBMA. So we might try the SMA 2000 also, along with DIBMA, and see also how these polymers are working with our system. So that was a very good remark.
Sure. I believe you will get less yield with deep mount. That is
Okay.
in my experience. If you want to grow like a 40 flask of culture and you just want to solubilize it, well, be careful. Is like make sure that it's worth it. You know,
Okay.
is a lot of work. You know, that's the thing.
Deep math seemed
Yeah.
interesting for us for the like very far-fetched goal of stereoselectivity and using circular dichroism because of no aromatic residues. But